Hello everyone, welcome back to Study Tips for IGCSE Biology. Today's topic is related to enzymes. Without further delay, let's begin. In this chapter, you will find out about how enzymes work as biological catalysts, how enzymes are named, about active site, substrates, and products, why enzymes are specific for their particular substrates, how temperature affects enzyme activity, why temperature affects enzyme activities, how pH affects enzyme activities, and why pH affects enzyme activities. What is biological catalyst? Chemical reaction can be sped up by substances called catalysts. Chemical reaction take place all the time within any living organism called metabolic reactions. Catalyst means that a substance that increases the rate of chemical reaction and is not changed by the reaction. And almost every metabolic reaction is controlled by catalyst, which is called enzymes. Enzyme or enzymes, protein that act as biological catalysts, meaning that they speed up reaction without being used up. A different enzyme is needed for each kind of food. Example, starch is digested to sugar maltose by enzyme called amylase. As you can see from this table below, starch is digested to sugar maltose by enzyme called amylase. Protein is digested to acid amino by enzyme called protease. Fat lipids is digested to glycerol and fatty acid by enzyme called lipase. And carbohydrate is digested to sugar by enzyme called carbohydrates. So a chemical reaction always involves one substance changing into another. In any enzyme, control reaction, the substance which is present at the beginning of the reaction is called substrate. And the substance which is made by the reaction is called product. How an enzyme works? An amylase molecule has a den in it called active sites. This has a shape that is complementary to the shape of part of starch molecule. So the starch, the substrate, fits into the active site of amylase, the enzyme, forming an enzyme substrate complex. And when the starch molecule is in the active site, the enzyme breaks its parts. And each enzyme can only act on a particular kind of substance. If you can see the diagram below, so this is the substrate starch. And this is the pictures that you see like a brown color is actually enzyme amylase. Enzyme, and this the darker side is the active side. So the substrate molecule starch in this case slot into the active site. If you can see the active site, so the second diagram show that the substrate starch it's in the slot in the active site. So each enzyme has an active site into which its substrate molecule fits exactly. So this enzyme is amylase and its active site is just the right size and the shape for the starch molecule. And you can see that this is the product produced. Okay, and the starch is split into maltose molecule. The enzyme is unaltered and ready to accept another part of the starch molecule. So this diagram is actually in sequence. First, second, and third. Property of enzyme. If you can see, what are the properties of the enzyme? Okay, all enzymes are proteins. Enzymes are made inactive by high temperature. Protein molecules are damaged by heat. Enzymes work best at a particular temperature. If you see from this diagram, human body usually work at about 37 Celsius. When you see that when the temperature is getting up and the rate of reaction is very high. At the rate of 37 temperature, you can see that the rate is almost at the optimum level. So, the temperature at which enzyme work fastest is called optimum temperature. Refer to a diagram A. So, this is the diagram A. Enzyme work best at a particular pH. Okay, this is talking about pH. pH is a measure of how acid or alkaline solution is. Some enzymes work best in acid condition and other work best in neutral or alkaline. Refer to diagram B. So diagram B is actually, it shows how pH affect enzyme activity. Like for example, if you see most enzyme, it work best at the particular pH, the rate of reactions. Okay, and enzymes are catalysts. They not change in chemical reaction which they control. It can be used over and over again. Small amount enzyme can change a lot of substrate into product. And enzymes are specific. Each kind of enzyme will only catalyze one kind chemical reaction, as you already studied before in the previous diagram. So for example, like starch is digested to sugar maltose by enzyme called amylase. You see a different kind of amylase for specific of substrate to produce a specific of products. Temperature and enzyme activity. Most chemical reactions happen faster at higher temperature. Molecules have more kinetic energy, moving around faster and bump into each other more frequently, meaning that when at higher temperature, enzyme is likely to bump into substrate more often than at lower temperature. So for human enzyme begin to happen from about 40 Celsius upwards. As the temperature increases beyond this, the enzyme molecules start to lose their shape. So the active site no longer fit perfectly with the substrate. So this situation we call it as denature. When the temperature at which enzyme work fastest is called optimum temperature. Below table, it shows that the optimum temperature for the certain enzyme to work for example, human, is 40 Celsius upwards. For the plants, is between 28 Celsius to 30 Celsius. And bacteria who live in a hot spring as high as 75 Celsius. pH and enzyme activities. pH solution affect the shape of enzyme, and most enzymes are their correct shape at pH 7. pH 7 is in a neutral state. When the active site no longer fit the substrate, it's called denature. When pH become very acidic or very alkaline. So, protease, for example, we take protease, enzyme from human stomach has an optimum pH 2. If you can see, this is because hydrochloric acid in our stomach work well in very acidic condition. With that, I would like to end my presentation. Thank you very much.